Good morning, everybody. I think we're going to start our session. And I just heard, and I'm not too surprised as well. Apparently 1,000 people attended the session virtually. So there's a lot of interest on this topic. So we're going to continue for day 2. And Naiffer Romero is going to be discussing the more USP perspective on discussing the pitfalls and challenges of nitrosamine confirmatory testing. So looking forward to your talk.
All right. Good morning, everybody. What a fun way to start the day, but talking about analytical chemistry. Again, my name is Naiffer Romero. I'm a principal scientist in USP. I've been lucky or cursed, depending who you ask, to be involved in this topic since very early on. But yeah, the idea today is really to a very high level point of view of some of the challenges that we are facing and we're seeing when it comes to testing of these impurities in the lab. So just as a reminder, what the mandate is from the regulatory guidance, we have to assess that risk, and then we have to go to the lab if that risk is flagged to conduct those confirmatory testing. We spent a lot of time yesterday talking about limits. So of course, if that limits become an issue, also the rigorosity of how we test this material is going to become really important to challenge our product or against that particular limit. So that's exactly where I'm going to focus during the presentation. What is really the expectations on this confirmatory testing? This is text extracted directly from the guidance. It's expected to use sensitive analytical methods with the appropriate limits of quantification and limit of detection. Some reference given to ICH in terms of how that method needs to be validated, what are the quality attributes of that particular method. Validation needs to be taking place in every single method that we develop. Again, every matrix is different, so there's really an opportunity to just use universal methods blindly. And of course, a reminder that we're all running against a deadline, which is August 2025. The very first version of the guideline gave us some sort of a parameters when it comes to those limits of quantification that was indicated. Later on, a little bit of the revision of that guidance provided additional information and gave us some additional information about what the quality of that analytical method should carry. So I'm going to highlight a few of these challenges that we have seen and we continue to hear from our stakeholders and the people that we're really trying to help navigate on all that. We're going to be talking about reference standard materials, which is a very controversial topic. Sensitivity issues, matrix effects, selectivity issues, artifactual nitrosamines that can be formed during the sample preparation, during the analysis, and again, a little information about some those interference and contaminations that can happen during sample preparation. So I'm gonna start with this one, quality and availability of reference standard materials. Again, if we're gonna test impurities in our product, we need a reference standard material to do so. So I'm gonna give you a brief second to look at what I have on the screen. And what you see here is, if you go into Google and do a Google search for this particular impurity, you're gonna find not one, not two, but a large number of suppliers of this particular impurity. And if you all have been immersed in this topic, you know that there's a common denominator here. And the common denominator is that, in fact, that impurity cannot be synthesized. The big question is, how then these materials are being offered commercially for labs to test materials. And in some cases, you can see there's even certificate of analysis for this particular impurity. So definitely a big wake-up call on where are we sourcing the materials that we're using to test this in the lab. In that particular case, the chemistry suggests, or is suggested that it will undergo these mechanism that you see there. But in fact, that's not the chemistry that will take place. The chemistry that will undergo, it's a completely different chemistry to form a completely different structure. It's not a nitrosamine what you get. So again, that's telling the way we design, or the people that are supplying this material need to be put a special attention and really careful on how this synthesis is designed and its control. On the right side, just a sample of what that NMR will look like, and you will probably see that for somebody that is just basically doing a very draft check of that structure, well, you're gonna see that those two spectrums look very similar. That's not the only case. There has been reports of some other cases where a particular impurity has been offered as a reagent, and where you can see, depending on the conditions of the chemistry of the synthesis, I can come up with a completely different compound. So again, the conditions how these syntheses are carried out are extremely sensitive to what the outcome of that material is. And to supplement this is also the characterization that it's carried out with these materials. Very strict characterization needs to be carried out to really confirm that the material that we have in the vial is actually what we expected to have. So I'm sharing here a couple of anecdotes. I'm not saying names here, but just sharing really anecdotes that we extract from our community, the nitrosamine exchange community, when we talk about reference standard materials. And in the case of the supplier A, again, these are just straight from anecdotes on the community. We see that some of the supplier, in this case, this supplier provide some C of A with that material. It provides some NMR and MS data. However, when that material is tested in the lab on the mass spec, you start realizing that the material and the fragmentation pattern, it really doesn't match what it's supposed to be doing. Further investigation, or try to investigate this with the supplier, end up in the supplier basically saying, no, I'm not gonna provide any additional data. The certificate of analysis is all you get. In the case of on the right side, supplier V, which another nitrosamine, yes, the supplier provided all the characterization data. However, once again, this material end up, once the user was trying to analyze samples, that material was in fact not that particular nitrosamine. And the way out for the supplier was, well, I'm not gonna provide any additional information, just don't pay me for what you just got from me, okay? So again, very critical, the kind of situations are emerging with the labs that are testing this. And on the bottom of the slide, it's a general statement coming from a CRO that does a lot of this testing for a lot of customers. And basically, I'm quoting them, it said about 50% of the customers provide their own reference standard materials for analytical testing. These materials tend to be relatively weak on characterization and quality. So again, this is a reality coming from a CRO that is doing a lot of testing for a lot of organizations out there. We knew this, we start realizing this, and again, in case of USB, you probably know USB nitrosamine, just the 1469, and again, I want to show you that we're more than just the general chapter 1469. We dedicated specific resources in the USB to really try to accelerate the development of these impurities, to make them available to our stakeholders. And again, carrying out with that, it's not just about designing specifically the synthesis of that particular impurity, but the characterization that carries out to produce these materials. So that it's in acceleration mode to produce more and more of the nitrosamines that industry is asking us to produce. Some of the priorities for the development of that are really evolving the same way that everything is evolving in this topic. We are relying heavily on that regulatory evolution and really the feedback from industry in these impurities that they are needing. So let me move on to selectivity issues. Selectivity got really flack for our analytical colleagues in the lab when this first issue emerged back in 2020. Between this dispute between a private lab and the FDA investigation, specifically with ranitidine, where we saw really the value of having that high resolution when was needed in terms of that interference. So again, going back and refreshing you a little bit of what was the situation, we have a particular solvent, DMF, which when we did all the analysis on mass spec was sort of an interference with the NDMA. So again, do we always need high resolution? Not always. But again, when these kind of cases emerge, when you might have an interference, well, that's where the value of having that high resolution system really comes into place. Matrix effects, some of the challenges when it comes to sample preparation. Again, matrix effects, we can break it down in sort of two categories. Those that, when the response of the sample get sort of biased based against the reference standard solution, and that's really due the metrics on the product or on the sample that we're trying to analyze, and some other ones that are potentially causing alterations on the ionization efficiency of my analysis that can definitely enhance or cause suppression effects on the analysis. So again, matrix is a very critical parameter when it comes to the analysis of these impurities. Again, we also have to put into perspective what are we analyzing. We're not analyzing an assay, 99%, we're analyzing parts per billion. So again, the matrix effect in this sort of analysis is a lot more critical than any other cases. These matrix effects get very evident when we're looking at different solubilities, the matrix, what is the solubility of those matrix, and the extraction process. How are we going to extract that analyte to really undergo that analysis? So it becomes really critical, the selection of the solvents that we use and the extraction strategy that we're going to use for doing the sample preparation. Even things like what is the size of the container where I'm going to do the extraction have to be put some thoughts on the design of that protocol of extraction. So again, when we talk about sample preparation and that extraction strategy, well, there are different paths that has been tested and has been designed depending on that matrix that I'm working against. We have the traditional total dissolution where we put everything on solution. However, that carries a series of pros and cons. We have the selective extraction where we manage the solvents that we're going to use to try to extract only the analyte that we're trying to test. Of course, this has to be weighed against that efficiency on the limit of quantification and detection of the method. Matrix precipitation strategies where we dissolve the sample, then we use an anti-solvent to precipitate the matrix so we can find ways to get rid of that matrix effect and just leave in solution where we're trying to analyze. Again, the design of this is not as simple as it sounds, but again, these are strategies that are definitely being used in the lab. And then we have other protocols that have been tested as far as solid phase extraction and dispersed liquid extraction as strategies to produce an effective solution to analyze. Again, it's not just about the sample preparation and also the instrumentation we're gonna use. Again, this is really, and you're gonna see this in one of my last slides, the whole concept of risk assessment is not only for the product, it's not only for identifying the risk of the nitrosamines in the product, but that same mindset needs to be bring up into the lab. We need to identify what are the risks that are raised when we are gonna analyze this sample. Again, what you see here is the famous case, or the infamous, I should say, case of the ranitidine, where we early started testing ranitidine via GC, well, we start generating nitrosamines in the injection pore of the GC because the high temperatures of that injection pore. So again, that risk assessment of what kind of instrumentation I'm gonna use and my strategy needs to be weighing even before we walk into the lab to start doing preparations. So this might look familiar in the kind of when we do risk assessment, but this is more probably focused, the same principles, but now focus on the lab. Have I identified amine source in any of my matrices or any of my sample preparation? Do I have nitrosating agents that can force formation of nitrosamines during the sample preparation? And again, what are the ideal conditions that I should probably use or manage to prevent formation of nitrosamines and then potentially get false positive during my analysis? So let me go back. Okay, so one of those strategies, and I like to highlight a couple of strategies that have been used to sort of bypass that risk of potentially forming additional nitrosamines during the sample preparation, is the use of what I like to call the sacrificing agents. And yes, I just made up that term. But basically, I'm adding a component to the matrix that will undergo nitrosation, so that separate the main analyte or the main nitrosamine that I'm trying to test. So that nitrosating agent that might be on the matrix, I'm somewhat entrapped by sacrificing another component to form that nitrosamine. Again, we saw that put into practice. What you see here is one of the EDQM published method for ramipril, where you actually see that they add specifically into the preparation pyrrolidine solution to basically do that, trap any nitrosating agent that might be on the matrix. Another strategy is the use of scavengers. We talked about scavengers yesterday, but yeah, it's the same strategy that we can use and leverage in the sample preparation to really trap any nitrosating agent that might be on the matrix, and we don't want additional nitrosamines to be forming as we do sample preparation. Fitze and others reported also the selection of the solvents sometimes can be really tricky when we're doing these extractions. And again, some of that design of the selection of the solvents and sometimes the wash of those solvents for the extractions can really mean the success on the analysis of the content of these nitrosamines. So what you see here, again, I'm not gonna go into the details, but you see that in the cases where that solvent was not washed from the sample, well, now you can see an increase on the formation of nitrosamine as the sample sits on the vial. Also, one thing that we need to consider is everything that touched that sample can potentially elevate the risk of forming false positive or produce other nitrosamines. Again, this is a good example. Elastomeric components that might be used during the sample preparation might carry out some level of risk. Specifically or generally speaking, comes with the accelerators that are used to produce some of these elastomeric components. And again, here you have an example of an accelerator that is used to produce that, and that accelerator leach out into the sample and produce a particular nitrosamine. And again, this was reported very early on when we started trying to link extractables and leachables to the topic of nitrosamine. Interference and contaminations are really everywhere. So this is something we need to consider as part of the analysis. What you see here, again, we're looking at that trace analysis, so the noise that we generate is critical on those signals, and how do we manage that? Well, what you see here on the right side, it's how solvents during the analysis can also affect that baseline. And again, affecting that baseline ultimately will affect that capacity on the or robustness of the analysis. So even the quality of the materials, the solvents that we use can be critical on the analysis of these impurities. And similar to that, really everything that touched the sample, right? We talk about the solvents, we talk about the components that we use to prepare the samples, the filters, the vials, everything that really touched that sample, we somewhat need to understand if there's a risk carried out by any of those components. We're starting to see now special lines of products or disposable materials specifically for nitrosamine testing, and that has to do with that leachable that might come out with those materials. Something really interesting on the bottom of the figure, you see a column. There has been also reports of nitrosamines being formed on the columns. So again, the chemistry of that column can potentially become an issue during the analysis. So again, interference and contamination of that sample becomes very critical when it comes to the effectiveness of testing the content of nitrosamines in that sample. I want to share a couple of resources. Again, going back to USP, it's not just about 1469. I want to share couple of the things that we have been really advancing since they're really early on, and I want to give credit to the entire nitrosamine team in USP because there's a lot of work that is behind these efforts and these activities that you see here. Yes, a lot of people know 1469 as an informational chapter that we put together, not as a requirement to the industry, but as a way to help industry navigating the whole issue of nitrosamine. Reference standards that are associated with that chapter, again, going back to how critical it is the quality of that material that we're using to test the materials. And then something that I'm very proud, some of you might know this, the nitrosamine exchange, which is the community that we built to really bring everybody together into the same table. Globally, so today we have more than 5,000 members. We just crossed the 5,000 members milestone, representing 80 countries from around the world. And really, this is everybody working on nitrosamine that comes. So they have a seat on the table to discuss what their challenges are, what are the problems that they're having, how we can resolve a lot of these issues. Every time a new regulatory update takes place, it's a great place to have that discussion about the impact that these changes are having, not just for a finished product, but also for suppliers. So there's a lot of the discussions of how these affect suppliers in the community as well. So it's really a very big round table that gives a seat to everybody that is somewhat involved on the topic of nitrosamine. In that particular community, we have a section, and we are in the process of revamping this, where we call the analytical hub. And really the idea of creating this, it was somewhat build a repository of analytical methods that people could use to start their development. This was not meant to be a method that you don't have to do your development, but at least it can give you a jump start when it comes to analytical development. And again, we're in the process of revamping so we can bring more and more of those methods that are out there into this sort of a database or repository that we have building in there. So some final remarks as for my presentation. Analytical methods with the appropriate limits of quantifications are needed to test whether drug products can meet the AI limits recommended for nitrosamine impurity. That's language straight from the guidance. So in order to accomplish that, we need high sensitivity methods. Again, we're looking at very, very low concentrations. Matrix effects, matrix interference can really be the breaking point of the analysis if we are not extremely careful how these samples and how these extractions are made. The selectivity of that method is critical. Again, we have had several experiences, and this is widely known and widely published, when the selectivity is not there, what the challenges are. Artifactual formations are critical as well, because it can come from the sample itself or from any component that I'm using as part of that sample preparation. Regulatory compliance, of course, it has to be there. The method has to be capable of fulfilling the requirements that are established to the test of these materials in a robust and precise way. Accuracy and reliability of that standard materials are critical. Again, my entire analysis is only based on that known component, which is the reference standard. So I better be using a good material that I can trust, that it has has been characterized correctly and it has the right purity reported. So that number or that challenge of my product, it's true what it is. So with that, I thank you for your attention. I know it was a really fast fly over this. You will have the slides available. And again, all I could say is I invite you to come and continue the conversation in the nitrosamine exchange. So thank you so much.
Yeah, thank you, Naiffer, for this talk. So the next talk is also going to be a little bit more also on the analytical side. It's Guozhang. He's going to talk about the extraction and quantification of N-nitrosoketamine. So.
Okay, I'm going to talk about the extraction and the quantification of N-nitrosoketamine from ketamine drug product using mixed mode solid phase extraction and LC tandem mass. So this is my disclaimer. And of course, the quality of drug is important. So we all have been fighting these nitrosamine impurities. So for the from previous talks, we know I think nitrosamine almost exists everywhere in drug substance, in the drug product, even in the excipient, even the air and the different place can affect the formation of nitrosamine. But the problem is, how are we going to detect, how are we going to quantify this nitrosamine impurities accurately? So that's what we're facing when we first get this project. So how are we going to detect this nitrosamine? So when we first got this project, then I did a Google search. So I didn't do ChatGPT because I reserved that one to solve the problems with my kids. So actually, ketamine is a very fascinating drug. So I read the literatures, ketamine is a non-competitive antagonist of the glutamate-gated cation channel receptor of NMDA that can block the calcium influx and therefore can prevent the neural activation. So ketamine has been proved as an anesthesia agent, and the S-isomer has been improved for antidepressant treatment. So the activity is inertial selective, and the S enantiomer is more potent than R enantiomer. So if you look at the structure, it has a binary ring and it has a secondary amine. So secondary amine, that's a source of nitrosamine. So the maximum daily dose of ketamine is 1,074 milligrams. And in the draft guidance, the N-nitrosyl ketamine is of category 5. So with a recommended acceptable intake limit of 1,500 nanograms per day. It's equivalent to 1.4 ppm or 14 nanograms per 10 milligram drug product. So I listed the table listed the composition of the reference of the listed reference drug catalog. It has 3 dose forms: 10 milligram, 50 milligram, and 100 milligram. It's equivalent to the amount of the drug substance ketamine hydrochloride. So exactly like I mentioned, to analyze this trace amount of nitrosamine impurity is quite technically challenging. Because we have to facing the large quantity of API and then try to analyze the nanograms of nitrosamine that's present in 10 milligrams ml drug product. The first I mentioned the challenge for chromatographic separation. Here, it doesn't mean we cannot separate nitrosamine from ketamine. It's because the large quantity of ketamine causes the peak tailing. So that's going to affect our analysis. And also we realize there's carryovers from the autosampler. People are going to argue, okay, you have the mass spec. Why do you worry about the LC separation? But the thing is, there's ion suppression. I think there could be possible different possible reasons. One is the ketamine going to compete to access to the surface of a droplet for gas phase emission, and going to compete for charges. And also because it's a weak base, probably going to grab a charge from nitrosamine. So how to solve these problems? We try to, improve the separation of reverse phase HPLC, and also try to divert the ketamine into waste first. But that didn't work very well. And other people tried Helic or other type of chromatography. The idea is to elute nitrosamine first and ketamine later. That way they can avoid the peak tailing of ketamine. We also tried liquid phase extraction. That didn't work well either. Finally, we tried solid phase extraction. That's proved to be a very reliable, very solid approach for us. So next, I'm going to explain what is this mixed mode solid phase extraction. If you look at the analytes, it has a large quantity of ketamine, 10 milligram per ml. It's a weak base, so it is positive charge on the acidic condition. And the trace amount of nitrosamine it's less than 10 nanogram per mil. But it's neutral, right? Because when you change from ketamine to nitrosoketamine, the nitrogen change from sp3 hybridization to sp2 hybridization. That become the base that become less basic, and also they can form this conjugate structure. It's a triton ion, so basically the charge be neutralized. So we choose the matrix, the solvent, that have binding ring has a sulfate ion. So that can have a mixed mode interactions. That means has a charge-charge interaction and a pi-pi interaction. So during the sample load and wash step, the surface, we use 0.1% formic acid. So the surface of the matrix is negative charge. So both the charge interaction and the pi-pi interaction are active. So when we load the sample, the ketamine will interact with the matrix through both the charge interaction and the pi-pi interaction. So that can be captured on the surface of the matrix. For the nitrosoketamine, that can also be captured on the surface through pi-pi interaction. Then in the elution step, we use the 0.1% formic acid in acetonitrile. So the nitrosoketamine, N-nitrosoketamine can be eluted out, but the ketamine remain retained on the matrix through the charge interaction. So in this way, we can collect the fraction and for our following analytical analysis. So this is a detailed step how we did this experiment. So we first prepare a sample. We take 1 ml of drug product. We added 1 microliter formic acid and the internal standard that's a deuterated N-nitrosoketamine. So we load the sample to the matrix. This matrix was preconditioned with 1 ml of methanol and then 1 ml of 0.1% formic acid in water. So we washed the matrix with 0.1% formic acid in water and then eluted with the 0.1% formic acid in acetonitrile. So this method is very straightforward and it's very reliable. So like I said, for the LC, actually it's quite easy. We use a very short gradient and the nitrosoketamine actually separated from the ketamine very well. Then for the mass spec, this shows the mass spectra of nitrosoketamine. So the insert figure that's the MS1 spectrum, 267, that's a monoisotopic peak. The +2, that's because its relative abundance is because it has a chlorine that comes from the +2 isotopic isotope of chlorine. It has three major fragment ions, 125, 179, and 207, and 2 minor fragment ions, 163 and 189. And then this one shows the the deuterated nitrosoketamine. Again, the insert shows the MS1, the same pattern as the nitrosoketamine, just with the +4 mass shift. And for the MS2, the same pattern as the nitrosoketamine, again with a mass shift of 4. So I gave the possible fragmentation pathways of nitrosoketamine. To the bottom, can either lose the N-nitroso group first, and then for the 207, 177, and 125 fragment ion, or it lose the water first, and lose forming the 189 and 163 fragment ions. So based on all the information, we identify the quantifier and the qualifiers of nitrosoketamine, and then we optimize the parameters for those transitions. So we established methods. Next, we did the method validation. So this method turns out to have the linear range from 1 to 50 nanogram per ml, and with a limit of detection of 0.5 nanogram per ml, and with a lowest limit of quantification of 1 nanogram per ml. So remember, the acceptable intake is 14 nanograms per ml from 10 milligrams per ml drug product. So that meets the need of our assay. Because we use solid phase extraction, so we measured the recovery, matrix effect, process efficiency, and accuracy at the 3 different levels: low, middle, and high. For recovery at all 3 levels is all above 90%, matrix effect less 10%, process efficiency about 84%, and accuracy about 96%. Those are pretty consistent among all 3 different levels. So we have this method, then we measure some samples. We measure the sample of the USP reference standard. We measure 1 API. We measure 1 drug product. For the USP reference, it's below detection limit. For the API, we detect 2.2 nanograms in 10 milligrams material. And for drug product, we get the 7.6 nanogram in 10 milligrams drug product. So this is a summary. So we established the methods using solid phase extraction and LC10 mass for quantification of unnatural ketamine. So we measure some of the samples. And of course, next step, we're going to investigate the formation of natural ketamine during manufacturing and in-use conditions. We want to investigate what's the cause of the formation of natural ketamine and try to find the mitigation approaches for this impurity. So I want to acknowledge the contributions of my colleagues and the discussion and guidance from the OPQ Nitrosamine Working Group. And I want to thank the organizer and I want to thank the audience for your attention. Thank you.
Yeah, thank you, Dr. Zhao, for that presentation. Very interesting. So the next talk, and actually the next 2 talks are interrelated. So if you see one, hopefully you do see the other because they're sort of very closely interrelated. You're going to see that. But the first talk we're going to talk is going to be from Jingyue Yang, Dr. Jingyue Yang from FDA, and she's going to talk about fragment API NDSRI fragments that result from impurities from the NDSRIs. And she's going to talk about measurements in certain select compounds, as well as some conversion rate assignments. So.
Good morning, everyone. As Andre mentioned, in my talk today, I'm going to share with you some measurement results for API fragment NDSRI, and then the conversion rate that we estimate from these results. So in my talk, the API fragment is interchangeable with impurity. So this presentation, this disclaimer, this presentation represents only the views of the authors and doesn't reflect any of the construed to represent any of the FDA's views or policies. And as Guozhang, the previous speaker, I also work for Office of Pharmaceutical Quality. And the mission of our office is to ensure every dose to the patient is safe and efficient, free of contaminations and defects. And by now, everyone in the audience kind of know this reaction very well. So we know that a vulnerable tertiary or secondary amines, they can readily react with nitrosating agent to form nitrosamine. And the nitrosamine is a possible or probable carcinogen. So a publication in 2023 kind of showed us that about over 40% of the API in the USP monograph and over 30% of the impurity, they contain either a secondary amine or tertiary amine, at least one secondary or tertiary amine group. So theoretically, many of these substances could form kind of like nitrosamine. So that'll be like a big issue. So far, the NDSRI formed from the API are very well recognized. So many of the effort have been put into to address the safety risk posed by API NDSRI. So Now the question is, how about the impurity or API fragment? So do they also form NDSI? So if they form NDSI, what is the safety risk posed by this impurity NDSI? So some may argue that impurity probably does not present as a very high safety risk because of their low level. So compared to the API, probably very low percentage of them are like impurities, so availability for the nitrocation reaction is probably as much as the API. And so the qualification threshold from the ICH Q3B kind of like shows that probably many of the drug product, the impurity are well controlled under at least 1.0% or something like that. However, the same group of the author for the publication that we just cited in the previous slide, they kind of like argue differently, showed a different picture. So in their publication, they showed that API fragment NDSI might be a serious like a safety risk. So there's a colorful box over here. So basically, the reading from this box is that if we have a drug product with a maximum daily dose of 1,000 milligrams, so even at a conversion rate of 1%, meaning 1% of the impurity converted to its NDSI, and if this impurities level is at 0.015% or above, so any of it will form the NDSI kind of like with acceptable, above the acceptable daily intake of 1,500 nanogram per day, the maximum currently allowed by the guidance. So this kind of like shows a very not that good picture for the API fragment NDSI. And to get a more accurate and comprehensive kind of estimation of the safety risk that may posed by API fragment NDSI, a group of the FDA researcher looked into the FDA's, I think it's global substance registration system, and they collected all the impurities with a secondary amine from this database, and they kind of like use a simulation to make the NDSI for all these impurities and calculate the risk, safety risk for all these NDSI formed from these like impurities. So you will hear this like interesting results from Xiang in his talk following this one and coffee break. So in their like simulation, 4 factors were considered, including maximum daily dose of drug product, acceptable intake of NDSI, as well as quantities of the impurity and the conversion rate from the impurity to impurity NDSI. So for the maximum daily dose and acceptable intake, those 2 values can be determined from the drug product and the NDSI associated with the impurity in question, and they are fixed values. And for the quantity of the impurity, a range was set for the simulation, and that range was set based on the qualification threshold for the ICH guidelines Q3B. And now the question is the conversion rate. We don't have much data or literature for the conversion rate of an impurity to its NDSI. So what kind of a range should we consider in the simulation? So to answer this question, we decided to take a laboratory study to get some like a real world like a drug product and to analyze them for the API fragment NDSI and get an idea about the conversion rate. For this purpose, we selected the 2 drug products, amitriptyline and ritonavir. The fragment NDSI from these 2 drug products were known, and in some ways they can represent some of kind of a worse than average case kind of scenario. So we're thinking that when we analyze API fragment NDSI from these 2 drug products and calculate the conversion rate, and this may represent some sort of like a upper end of the range we look into for the conversion rate. So that's kind of like what I'm going to share with you next is our results we get from the analysis of these 2 drug products. So I will first start with amitriptyline. As you know, that most of you know that amitriptyline is antidepressant medication. It is available only in the dosage form is available only as a tablet, and the dosage strength, six dosage strengths ranging from 10 milligram to 150 milligram, and maximum daily dose is 300 milligram. So for this purpose, we analyzed 14 lots of the drug products. So those 14 lots represent like 6 manufacturers, denoted here by letters A through F. And among these 14 lots, 4 of them are expired. The earliest expiration year is 2021. And we also get like 2 different dosage, 150 milligram and 10 milligram from manufacturer ABC. So the reason to get it is to analyze both dosage size. We kind of like wanted to have some idea that whether dosage strength or size of the tablet could have an effect on the NDSRI kind of formation. And amitriptyline is like tertiary amine. So theoretically, it can undergo nitration to form 3 kind of like a nitration product, with each product with like each of this group connected to amine, connected to the nitrogen to be replaced by a nitrosyl group one at a time. And because amitriptyline has a 2 methyl group, so at most it has 2 nitration product. One of them is NDMA, and this is a very well-known probable carcinogen. So it has a acceptable daily intake of 96 nanogram per day, which is translated to a limit of 0.32 ppm. And then the other one is N-nitrosyl dismethyl amitriptyline. And from the CPC calculation, this compound has acceptable intake of 26.5 nanogram per day. So that is translated to 0.88 ppm limit. And the dismethyl amitriptyline, which is secondary amine, so this is a kind of like a known specified impurity in amitriptyline with a specification limit of less than 0.2% in the drug product and less than 0.15% in the drug substance. So the formation of the nitrosyl dismethyl amitriptyline is probably not the nitration product of the amitriptyline, which is tertiary amine, but instead is probably the nitration product of dismethyl amitriptyline, which present as a impurity in the amitriptyline product and also a secondary amine, which has far easier reaction than the tertiary amine. And similarly, the NDMA probably also come from the nitration of dimethylamine instead of amitriptyline. And dimethylamine, or DMA, is not really included in any of the as a specified impurity, but we do find DMA in all the amitriptyline drug products that we screened. And we developed like 2 LC-MS methods and to analyze this like analyte. And both of the methods start with extraction of analyte from the tablet by methanol, and then the sample were analyzed by LC-MS, first subjected to the chromatographic separation by reverse phase LC, and then ionized by mass spec and analyzed by high resolution mass spectrometry. And we did the quantitation using the external reference standard. So the first method is used for the analysis of the NDMA and the nitrosyl dismethyl amitriptyline. And for this method, we have like a limit of quantitation about as a 0.04 ppm for both analytes. And then for NDMA, the range is up to 0.3 ppm. And for the nitrosyl dismethyl amitriptyline, the range is up to 2 ppm. And the second method is used to analyze the dismethyl amitriptyline alone. And for this method, we have a limit of quantitation of 0.02%. And the upper limit for, or the up end of the range for this method is 0.2%. So to calculate the conversion rate, we take the concentration of the liposoamine and divide it by its precursor. And the value is in the unit of weight percent, represents that how much percent of the impurity being converted to its NDSRI. So first I share the screening results for the dismethylamtryptyline and liposomal-dismethylamtryptyline. Liposomal-dismethylamtryptyline, the values are represented by the bars here. So as you can see that we have the highest value detected is 0.87 ppm, and the lowest is, it's kind of like a wide range, and then lowest is only 0.007 ppm. And for this kind of 14 lots we tested, 11 of them come above the limit of 0.088 ppm, which is showed here by a red line. And we do see like a very high level liposomal-dismethylamtryptyline in the expired lot, which is marked by the red star over here. And these results kind of like grouped by the manufacturer. So when you look at like each manufacturer, you can see there's some difference there. So particularly, we noticed that for the manufacturer C, all the lots we tested, they have a very low level of NDSRI, and also, yeah, very, very low. So, and for just mentioned, so for manufacturer A, B, and C, we also tested both 150 milligram dosage strength and 10 milligram. Just by like a visual inspection, we can see that the value in the NDSRI in the 10 milligram dosage strength, which represented by the line bar over here, and always kind of like higher than the 150 milligram dosage strength for like manufacturer A, B, and C. So the results for the dismethylamtryptyline represented by the orange dots over here. And the range for the value is from 0.026 to 0.077%. It's not much difference here. And other than the data from one lot of the manufacturing B and manufacturing F, you can see that the range of the dismethylamtryptyline sort of like between 0.3% to 0.4%. And it's pretty much consistent across different manufacturing or like expiration date. And similarly, like there's no kind of like correlation between the dismethylamtryptyline level and its NDSRI form. So with these two kind of results, we calculated the conversion rate for the conversion of the dismethylamtryptyline to nitroso-dismethylamtryptyline. And these values are represented by the red diamond in this graph. And along with this red diamond, this conversion rate, we also listed the like test results for the NDSRI, and they are kind of like ranked from the highest to the lowest. So from this graph, we can see that the conversion rate from for the conversion from dismethylamtryptyline to nitroso-dismethylamtryptyline is between 0.002 to 0.24%. Again, it's kind of like a wide range. And again, we see that we observed high conversion rate with expired lots. And for the current lot, there are 10 of them, and they all below 0.07%. And similar to the NDSRI results, we see like particularly for manufacturing C, they have a very low conversion rate compared to other manufacturers. And then the level of the range of the conversion rate is correlated with the NDSRI level. the other, as mentioned, the other nitrosamine possibly present is NDMA. So we're also looking into the NDMA levels in the amitriptyline. And we found that the highest level we found is 0.098 ppm. And in some lots, we could not detect them. And these lots are all from manufacturer C. And all of the NDMA levels in all lots we tested, they are below the limit of 0.32%. And again, like a relatively high level of the NDMA can find in the expired lots. And the level of the NDMA seems like have some correlation with the liposyl dismethyl amitriptyline levels. So for the DMA, we didn't have time to validate the method. So we basically just give a little bit of like a estimation of how much DMA we see in this drug product. So the level of DMA in this drug product ranged from 50 to 150, 145 ppm. Again, that's as an estimation. And then we use this estimation and NDMA value to calculate the conversion rate. So the rate here is from 0.002% to 0.1%. So this is sort of like consistent with the previous conversion rate from the dismethyl amitriptyline to liposyl dismethyl amitriptyline. So the second drug product I'm going to share is ritonavir. And ritonavir is an antiviral medication. It is also used as a pharmacokinetic booster through its deactivation of some drug metabolizing enzyme. And ritonavir can be available in like a tablet, capsule, powder, oral solution. As a tablet, only one strength is available, like 100 milligram, and 80 milligram per ml is available for oral solutions. The maximum daily dose for ritonavir is 1,200 milligram when it is used for the HIV treatment. And ritonavir is also part of this popular, Paxlovid, the popular COVID treatment. And when used for this purpose, the maximum daily dose is 200 milligram. So for ritonavir, we analyzed the 4 lots of the drug products. All of them are tablets and have a strength of 100 milligram, and all of them are current lot. So with a look at the structure, you know that the ritonavir can have a very complicated impurity profile. So one of the degradation paths for ritonavir is a hydrolysis of amide bind, shown by the red part of the structure. So with this degradation, secondary amine, like a degradant, can be present in the drug substance or drug product. And then this secondary amine fragment will be converted to its liposyl form, which we have a short name, NITMA. So the presence of the NITMA in ritonavir was like a matter of concern from a recent FDA public announcement. So for NITMA, by the CPCA calculation, we have like a 26.5 nanogram per day for this impurity, so it's pretty low. And based on the maximum daily dose, we can have like a limit of the 0.202 per ppm or 0.13 ppm. So similarly as amitriptyline analysis, we also have 2 methods to analyze that impurity and then the fragment and the SRI. So for the impurity, the limit of the method is, the limit of the quantitation is 0.025%, and the range goes up to 0.25%. So for NITMA, the limit of quantitation is about 0.25% or 0.25 ppm, and it goes to 5 ppm. So here are the table of the summary of the results for the ritonavir. So for the NITMA, like the value goes from 0.27 or 0.28 ppm to 0.8 ppm, and the fragment also, goes from 0.06 to 0.3%. So it seems like no correlation between those two. And then when you calculate the conversion rate, it goes from 0.01 to 0.1%. So the upper range of this conversion rate is kind of like in the same scale as amitriptyline. So as a summary, that in this study, we looked at two drug products, amitriptyline and ritonavir, to make assessment of the conversion from the fragment to fragment NDSRI. So for amitriptyline, we have estimated the conversion rate range from 0.002 to 0.24%. And for ritonavir, it's 0.01 to 0.1%. Amitriptyline, we analyzed 14 lots of them, so we kind of observe some trends regarding this conversion rate. So we do see high conversion rate for expired lots. And then for the current lots, they are kind of below 0.07%-ish. And it does show some manufacturing difference. One particular manufacturer has a very low conversion rate compared to other manufacturer. Even they work with same impurity, impurity NDSRI. So with that we kind of have an idea of what conversion rate we really look at for this simulation. And this will go to the next talk by Xiang, who presents this very interesting results after coffee break. So this work was initiated by Andre Rohr, together with Matt Vera, Dan Berger, and Joanne Chang from Office of Pharmaceutical Quality Assessments. And the experiment was executed at the Office of Pharmaceutical Quality Research by Tim Guozhang, Yan Wu, Dia Shekilaya, under the leadership of David Carey. Thank you.
So, thank you, Jan, for this very interesting talk. And then there's going to be a subsequent talk that's sort of, we do actually simulation to see how much we would predict we would get this in our drug product repertoire. And this conversion extent is very critical, variable in that simulation that we had to experimentally determine. So next talk, as I mentioned to you, will be talked by Dr. Xiang Yu, and the talk is going to take a lot of information from the prior talk, and we're going to do some simulation modeling based upon our repertoire of impurities to sort of have an insight about this impurity, API impurities, these resulting in NDSRIs above the CPCA. So, and his title is Fragment Nitrosamine Risk Based Upon Quantity, Extent of Conversion Rate, and CPCA, automated calculations. So.
Thank you, everyone. My name is Xiang Yu. I'm working for the immediate office of pharmaceutical quality assessment 1 on the OPQ, say the FDA. My main responsibility for the office actually is doing programming. That's why I'm here giving this presentation about the NDI evaluation of API fragments based on the quantity, extent of conversion rate, and the CPC approach. It's pretty simple, straightforward simulation, but the results are pretty interesting. So the presentation will include 4 parts. First, the part is the background introduction about NDSRI, API fragment, and NDSRI risk, the GSRS database, where the data comes from, the CPCA approach for AI limit evaluation, and the risk formulation for our risk simulation study. The second part will be the data preparation. I will explain how we prepare the data for this simulation study. And the third part is the simulation study. It will be one individual impurity study and overall evaluation. And the last part is the conclusion. So NDSII is nitrosamine drug substance-related impurity. FDA has a specific website to list all the confirmed NDSII with AI limit. And this simulation was done in the summer of this year. As of July 2024, this website lists 264 NDSII impurities from 227 APIs, with AI limit given by the CPCA approach. And there is 4 NDSII limits at that time was based on the compound-specific data or read-across analysis. So we can make 2 conclusions. First, there is very limited real-world toxicity data to support the risk evaluation of impurities. And second, if we look at the structure about these 464 NDSII impurities, we will know they are very close to the API itself or directly derived from API itself. Therefore, in September 2024, FDA further revised its final guidance of NDSII. One of the updates is to address the potential risk of API fragments, which means impurities that are not structurally close to the corresponding APIs, but may expose a high NDSI risk. And this is the classic NDSII scenario. So API itself may have the secondary or tertiary amine group and can be directly nitrosated to form NDSII. But in a lot of cases, API itself may not be nitrosated, but its fragment, either from the process or from the degradation, may be nitrosated and form NDSII. We call it fragment NDSII. And this is a confirmed case. Cyprotoflexin API, it has a secondary amine group and can be nitrosated with the NDSII currently is on the FDA's AI website, while the piperazine, which can be the starting material or the process impurity of this API, can also be nitrosated to form NDSII. While this impurity structure actually is not listed in the FDA's website. Our impurity data comes from GSIS. GSIS is the FDA's global substance registration systems with UNI for ingredient in the medical products and deployed by FDA in 2006. It's a collective effort among FDA, NIH, and EMA. It has multiple data resources, including but not limited to the pharmacopoeias like EP, USP, and the public chemical database such as PubChem. Also, there are more and more impurity data from like applications such as DMF, NDA application, and NDA application will be gradually put into the database. This because GSIS has a very sophisticated search and data filtering functions. It makes us feasible to perform the simulation at a relatively larger scale with very low cost. And the GSIS is a government-funded project, therefore its source code is publicly available. Cannot move, okay. So if we attend yesterday's talk, we should well know the CPCA approach. CPCA approach is a structural activity relation-based method assigning a nitrosamine to 1 to 5 categories, each with a corresponding AI limit with the table listed here, and reflect the predicted genetic potency. It considers the impurity structure features, including the alpha hydrogen, tertiary alpha carbon, carboxylic acid group, and other structures. For the detail, you can look at the FDA's guidance as well as the research paper for the development and validation. The CPCA approach overall is a very conservative approach with acceptable negative false rate. That's why FDA and other agencies use it to evaluate the risk of NDSII impurity. So for our study, we use a risk formula to evaluate the risk of individual NDSII impurity. The risk equals the quantity times the conversion times MDD and divided by AI. The quantity is the percentage of an impurity with secondary amine groups in the drug product. Conversion is the empirical conversion extent for that impurity to form NDSI in the drug product. MDD is the maximum daily dose of the corresponding APIs associated with the impurity. And in our simulation, we only use MDD records with units like milligram, microgram, and nanograms. And the AI is the acceptable intake according to the potassium category predicted by the CPC approach. So the meaning of the risk score in our simulation is pretty straightforward and easy to understand. If the risk value greater than 1, for example, 1.5, it means 150% of AI limit. That will be definitely above the FDA recommendation and will bring concern. If the risk value is below than 1, for example, 0.5, it means it's only 15% of AI limit, then the risk will be considered to be low. So we use the impurity data from GSRS. For first step, we directly retrieve all the impurity labeled as impurity in the GS database. Then we use the in-house CPCA program developed by FDA GSIS group to filter the impurities with secondary amine groups and calculate the AI by CPCA. After that, we further use GSIS to find the impurity with the corresponding drug substance and do necessary data cleaning. And we generate a data set of 1,933 impurities from 571 parent substance. Because these impurities may come from products like cosmetic product or like a drug product approved for animals, then we further screening generate impurity API MDD alignment based on the CDA's MDD database for the human drugs and form a final data set including 1,279 impurities from 321 drug substances. One thing I should mention here, we compared this API with the API listed in the FDA's AI website, and we noticed more than 90% of these APIs actually are not included in the website. It means this data set actually is well suited for our simulation purpose. And this is the formula for our risk evaluation. And in this study, we take the range of API quantity from 0.01% to 1%, which are frequently observed in current NDA and ANDA submission. And the range of conversion is set up to 0.001 to 0.5 based on the current available experimental data. And as I mentioned before, this study will include one individual simulation as well as the overall evaluation for all the impurity sets. For the public concern of speech drug product, we had the product information for this individual study. We know for this impurity, it has the potassium category 1 with very low air limit, and we know the drug product MDD value is 418 microgram per day. This 3D plot is the risk value for that impurity according to the variation of quantity and the conversion. And we give a table to represent how the risk value change based on the quantity and the conversion. With the yellow color highlights the rows that the combination of quantity and conversion resulting in the risk value pretty close to 1. And we can see if the quantity and the conversion just above this small number, then the impurity level in the drug product will be above the FDA's recommended AI limit. For the overall evaluation, we can see this is a 3D plot for the percentage of impurity with risk greater than 1 after changing the quantity and the conversion rate. And we can see at, how to go back? Huh? Click what?
There's a red one.
Red one? This one? No, it's go further forward.
Do you want to go back?
Yes, go back.
Is it going forward or back in your slide?
Okay, yes. Okay, thank you. So, if we look at the commonly said the impurity level of 0.2% with conversion rate 0.1, we will see the percentage of impurity will be greater, with risk greater than 1 will be 32.12%. And the conversion rate of 0.1 is the median value that are observed from current studies. And if we look at the distribution of the risk score among these impurity sets, with the quantity 0.2% and the conversion rate 0.1%, we can see this long tail distribution. And we can see that more than 1% of impurity with risk score greater than 100. Actually means this set of impurity actually has a very high risk. And this is the log plot of the how risk distribution at the quantity 0.2% and the conversion rate 0.1%. We know that MDD is a fixed value based on the drug product or API itself. AI is based on the impurity structure itself. So these two are fixed variables. Quantity may change according to the MDD value based on the SHQ3B. Therefore, because for this impurity from GSS, we don't know whether it's a process impurity or the degradation product of the product. That's why we change the API quantity based on the SHQ3B QT limit as well as the MDD value for the drug product. And change the conversion rate from 0.01% to 0.5%. And this is update the percentage of impurities with risk greater than 1. At the quantity 0.2, we can see actually that the number of impurity with risk score greater than 1 actually is pretty large. Of course, in the real situation, the result may be much better because normally the impurity level will be far below the SHQ3B. And also in this simulation for the MDD, we didn't differentiate the solid product from the liquid product because liquid product normally the risk is much lower with a very low conversion rate. So based on the observation, there are so many impurities. We should separate impurity with the high risk group and the low risk group. For the high risk group, we normally should consider drug product with high MDD values. Also, the impurities with the CPI category, normally I can 1 or 2, 3. And it should be noticed that it doesn't mean like category level 3 is have relatively low risk because in our simulation study, we noticed actually the impurity with the highest risk score is from the category 3 because of very high MDD value. And we also check the drug product dosage form. For example, if the drug product is in solid oral dosage form, like a tablet capsule, typically it will have higher conversion levels for these impurities. Also, for the low risk situations, normally the drug product should have pretty low MDD values. Category by CPC approach normally is 4 to 5, and typically liquid product may represent slightly lower conversion rate. So this is the conclusion we can make from our simulation study. The secondary amine impurities, or the API fragment with secondary amine groups, should be considered as a possible source of NDSRI in the drug product. So controlling the secondary amine group impurities to a minimal level may effectively limit the fragment NDSRI formation in the drug product. As I mentioned before, in our study, majority of API associated with impurity contain secondary amine groups are not listed in the current FDA's AI website. It means currently a lot of impurities with secondary amine groups that are not structurally similar to the API itself are off our radar at this moment. To establish NDSR control strategy, we should pay attention to the drug synthetic process to very carefully limit the level of this API fragment impurity with secondary amine groups. Maybe SHQ3A IDQT may not be enough. And we also need to optimize the drug product formulation to reduce the risk. It means not only optimize the composition of the drug product, but also optimize the quality of the excipient itself. And finally, I need to thank Dr. Andrew Rao, who is the leader of this project, also the leader of the NDSI working group on the OPQ, Dr. Tyler Pia, who provided support for this simulation. He is also the developer of GSI database as well as the CPC approach. And Dr. Joanne Cha, Dr. Matthew Vera, and Dr. Dan Berg, who provide valuable suggestions, and they are also the members of the kind of working group. Thank you very much.
Yeah, thank you, Shane, for the talk. Very, very nice presentation. And so the last talk for the session is going to be from Dr. Nitish Sharma, and his title is NDSRI Analysis, Risk Assessment Methodology.
Yeah, good morning, everyone. I'm Dr. Nitish Sharma, working as an assistant professor in Department of Pharmaceutical Analysis, National Institute of Pharmaceutical Education and Research, Naiffer, Ahmedabad, India. So today I'm going to discuss about NDSRI analysis, risk assessment methodologies. So the problem statement in today's scenario in any of the pharmaceutical organization is organic synthesis or formulation department. When they are developing their formulation or API strategies, they require possibilities of the NDSRI in the API of the formulation at very initial stage. Now the responsibility comes under analytical development department that how the NDSRI quantification will be possible without having an impurity standardization. So we have optimized a strategy to generate the NDSRI in the API and the formulation. So I will not brief this introduction because yesterday onwards we are many lectures we have listened about NDSRI. NDSRI are carcinogenic, mutagenic, and the source of NDSRI can be many. So yesterday also speaker discussed about the environmental condition which can also contribute for the NDSRI. So in 1978, early WHO has published a nitrosation assay procedure, that is a NEP test. In the NEP test, generally they prefer to use NaNO2 and an acidic environment. The pH will be around 3 to 4. By using this procedure, one can generate NDSRI or the nitrosamine impurity in the solution form. Now there are some limitations for this method. Aqueous acidic basic condition leading to many artifacts due to the hydrolysis, degradation, and side reaction. Second thing is most of the drugs are BCS class 2 and class 4 are not soluble in the aqueous form solution. So we need to optimize this, and the reaction mechanism of the NAP test is quite described earlier. So I will discuss, we have published the modified NAP test in the Journal of Pharmaceutical Science, and I'm going to discuss related to this into my today's presentation. So instead of using NaNO2 and HCl, just we need to add a tertiary butyl nitrite, so to 2.5 millimolar of API solution and 25 millimolar tertiary butyl nitrile solution, just vortex it, keep for incubation for 37 degrees centigrade for around 45 to 50 minutes, and perform the HPLC or LCMS analysis. Because we are able to generate a huge amount of NDSRI, so we can initially we can perform the analysis by using HPLC as well. So it is compatible with all of the chromatographic solvent like Snider, like methanol. We can use Snider is to water ratio. We can use water as well. In some of the trials we perform with DMSO as well, and it is quite suitable, and we observe the NDSRI impurity. So in tertiary butyl nitrite, the NO will be released and it will convert into tertiary butanol. So less artifact will be generated during the reaction. Now, third test which we have included in our final risk assessment methodology is we will add NaNO2 either in the solid form or in the solution form in the drug formulation, and kept the drug formulation for the stability for 7 days, 15 days at the real-time storage or accelerated condition, and we perform the analysis using LCMS. So intentionally we are adding nitrosation agent in our formulation or API. So the workflow of the conformative test just optimize a simple and responsive nitrosation technique for the synthesis of NDSRI. Then we can perform the characterization of this NDSRI by using LCMS, HRMS, or NMR. So the next step is we can determine the purity of NDSRI by using orthogonal technique. In solution form also there are several articles available for the reverse gradient CAD or LC CAD. We can determine the RRF of the NDSRI by using simple HPLC method or by LCMS method at a higher concentration level, and that can be correlate. Then risk assessment of the degradation product having the ability to form nitrosamine in the marketed formulation. So once we understand the retention time and related retention time of NDSRI, so it is very easy now to access the formulation and API sample, and we can check the formation of NDSRI in our real-time sample. So this can be a decision flow chart. As per me, this is my prospect, so it's not a conformity. So we need to identify first a drug or the impurities related to drug may have a secondary amine. This, if it is not, then no need to prepare any risk assessment report. If yes, we can perform NAT test, modified NAP test, and the formulation with NaNO2 spike, that is a conformative test. Now, any peak observed other than API, then yes, then we need to perform the HRMS analysis, then we need to characterize that impurity, and if an NDSRI has been generated, then we need to access the formulation or API stability sample analysis. If not, then we can give a justification based on the chemistry and reactivity of the compound. Now we did comparison in our laboratory with the NAP test and modified NAP test, how much NDSRI has been generated, and that can be correlated. So we selected a 5 API including hydrochlorothiazide, desloratadine, first 2, where we have observed no artifacts or no additional impurity in the NAP test. So by using NAP test, we are able to convert around 50% of the compound into the NDSRI. While using modified NAP test, we quite achieve up to 90%, that is 89.8% in acetonitrile and 96.6% in acetonitrile for the desloratadine. In acetonitrile, in 1:1 molar hydrochloric acid 9:1 ratio, 99.3% compound got converted into the NDSRI. And no secondary degradants has been. Just you need to evaporate the acetonitrile and the final compound is ready. So you can characterize it by using orthogonal technique, and you can determine the potency of that compound. Now this is quite difficult. Dabigatran, we performed the NAP test, and only 12.8% of the dabigatran is able to convert in the NDSRI. So this is quite difficult to isolate because 5 other degradant peak has been generated in this. So it's quite difficult for the analytical scientist to isolate this compound with the complex reaction mixture. Now, when we perform the modified NAP test, we observed the 93.5% of the NDSRI generated with 2 additional peaks. That is more than 1%. If you go with the Snider to water, 80% of compound has been converted into the NDSRI, and only 1 degradant product has been formed. So that is the benefit while we are using modified NAP test. Now the next compound we have used a tamsulosin. In tamsulosin also 15.3% of the NDSRI has been generated. While modified NAP test we are able to convert around 70% with 0 other extra additional peaks. So it is quite easy for analytical scientists to isolate this compound and make a working or reference standard. In the fluoxetine, only 1 secondary amine functional group is present, and there is no any other degradation peak has been generated while using a modified NAP test. So 99.4% compound got converted into the NDSRI. While in the NAP test, only 40% compound got converted into the NDSRI, and other peak, we got 1 degradation peak as well. I will not go through each and every chromatogram. This is supporting data already. This article we have published. You can refer it. I have added these chromatogram and spectra for your reference. You can refer my slide. So we performed the HRMS analysis, and we found that nitrosation happened on this secondary amine. This is the MSMS chromatogram, and this is the MSMS fragmentation pathway where we predict that this nitrosation happen on this condition. Now, someone asked yesterday that this nitrosation can happen on the nearby carbon or any additional element. So we can perform the hydrogen deuterium exchange. So that will give you the clear-cut idea where nitrosation happen, because the exchangeable hydrogen which is attached to the nitrogen are 3 only, and in NDSRI we observed only two mass unit shift, so 324 to 326 point something. So it defined that nitrosation happen on this nitrogen. The other nitrogen which is near to the sulfur, it is having more electron withdrawing group, so less probability to form that. So this is the chromatogram of decelloratadine, where we can observe that decelloratadine NDSRI impurity has been generated in modified NAP test. Well, we perform the HRMS analysis and MSMS spectra. The same we have reported in the article. Dabigatran in NAP test, we can see many additional big peaks, which is not easy to isolate the NDSRI while using NAP test. So we can perform the modified NAP test, and around 70% compound we can isolate. Tamsulosin, in tamsulosin also, I think only 1 secondary amine is present, and the more probability to form a nitrosamine here. So this also we prove with the MS and MSMS spectra. Now, someone asked me about the reproducibility of the modified NAP test. So we perform the modified NAP test reproducibility by using a complex reaction mixture, that is dabigatran and tamsulosin. And what we observed that it is quite reproducible. We perform this analysis on a different day by using different HPLC system and a different analyst. And we found a quite reproducible result while generating the NDSRI. Now in the dabigatran solution, we intentionally added this known quantity of NDSRI and API in the solution form. So we perform the reverse gradient LCCAT, and we determine the concentration of the impurity and the compound. Approximately, it's quite matching. So many of the articles are published related to this. So you can utilize that. And you can perform that analysis. Now the last molecule, a small molecule, is the fluoxetine. Fluoxetine only have 1 secondary amine. There is no other possibility to form the NDSRI. So we have used strategy 2 and strategy 3, and we observed that the compound has been converted into the NDSRI, and we predicted with the HRMS and MSMS data. Now move further, this strategy we have applied to the bigger molecule, or we can say is the medium size or the macromolecule. So it's a glycopeptide we have selected where the secondary amine has been introduced with the modification of the glycopeptide. So unluckily we can say that only vancomycin formulation is available in the Indian market, and oritavencin and dalbavancin is not yet available. So we have prepared an in-house formulation by using a formulation published by the innovator company. So vancomycin, only one nitrogen secondary amine group, functional group is present, that is additional in the side chain. Dalbavancin also we have an additional single secondary amine functional group, and in oritavencin we have a 2 additional secondary amine functional group. The upper one is having a restricted steric hindrance, so less propensity to form a nitrosamine NDSRI impurity, while on downside secondary amine is more vulnerable to form a NDSRI that we have predicted based on the MSMS spectra. Now in the glycopeptide, we optimize only strategy 2 and strategy 3, that is modified NAP test and the confirmative test. We have added, as these all 3 formulations are lyophilized formulation, so we have added NaNO2 in the solution form, and we kept for the stability. And we found in the formulation sample as well, this NDSRI has been generated. So we don't know the nitrosation, that nitrosating, ions are present in our formulation or not, because we do not have real-time sample available. So we have added intentionally NO in the reaction mixture and when performed the HRMS analysis. So this is HPLC chromatogram, the NDSRI we observed a doublet peak. We assume that this is a syn and trans isomeric form of the impurity, and we confirm this with the mass spectra. So for peptide and glycopeptide, I should let you know that the mass interpretation is quite different. So luckily in glycopeptide, only side chain modification happened during the MSMS fragmentation. No peptide chain will be affected during the fragmentation. So we have observed around 1816.622 to 0 mass of the dalbavancin, and NDSRI it's quite 1845.6130. So monoisotopic mass for this compound is 908 and 922. When we deconvolute this, it will convert, and we observe the ppm error for less than 5 for all, because by structure also we can predict the mass of NDSRI impurity. So we predicted that MSMS spectra in a different way as well. So the slight modification happened, and in API as well as dalbavancin and NDSRI. So this NC bond will break, and it will form 892 fragment ion, that is a signature ion. Other fragment ion also we have observed by removal of this sugar moiety, and it will convert to OH. So that also will convert in the secondary ion. So many fragmentations we have predicted, and based on the MSMS fragmentation, we come to the conclusion that nitrosation happen on this secondary amine. So the second compound is the vancomycin. In the vancomycin also we have observed NDSRI generation. So we use vancomycin capsule. It is available in the Indian market, and vancomycin injectable formulation that is also available in the Indian market. So according to the nitrosation process, we observed that 28.9 around mass difference we have observed for the vancomycin and NDSRI. This is a signature ion where this NCO bond will break, and this structure having only secondary amine. So other part will remain unaffected during the MSMS fragmentation, and we predict that this is the only possibility, and the mass ppm error will be less than 10. Now coming to the third molecule, that is oritavencin, where we have a 2 secondary amine functional group, and we perform the HPLC analysis while generating the NDSRI, and we observe the doublet NDSRI peak in this as well. So the total ion chromatogram, extracted ion chromatogram for oritavencin, and extracted ion chromatogram for oritavencin NDSRI, we observed the mass with the ppm error less than 5. So it predicts that this is happening being on the downside secondary amine functional group. So this also we predicted based on the MSMS fragmentation, and it is quite reasonable that this nitrosation happen on the downside secondary amine functional group, and by theoretical aspect also it predict that upper one will be strictly hindered. This all data we have published in this manuscript. This is already accepted in the supplement data. All the mass spectra and MSMS fragmentation we have captured. You can refer this article. This is accepted, not yet published, but it is about to publish in Journal of Pharmaceutical Science. Now it is a conclusion for the secondary amine in the glycopeptide due to semi-synthetic modification that can form the NDSRI and a paradigm shift for assessing NDSRI from a small molecule to the peptide and glycopeptide. Now, if someone asks me, yesterday also I have listened, the main objective of this workshop is NDSRI risk assessment is required for if any change in the excipient or API grade change. So this example is not yet finally concluded by my research group, but yet I think it's somewhat relevant to this topic. So I have added this. So NMR and other things are pending, but based on the HRMS data, I am trying to justify this. So we all know that secondary amine and formaldehyde will react. This is multiple publications are available for this reaction, and this secondary amine can react with the formaldehyde that will form a hemianimal and then will form NDSRI. The second reaction mechanism, if you use a histidine amino acid and lysine amino acid, that can cyclize and that can form a 12 addition mass impurity. So many of the protein molecule, this impurity has been already reported. Now if this happen, this convert this secondary amine functional group. Now, additionally, we have published one more article that is related to a peptide molecule, reactivity of N-terminal histidine peptide with the excipient or impurity of the excipient. So we predicted this earlier structure. I need to go back, one slide back. Okay, so in this we have reacted the API with formaldehyde and form this structure that is listed in this slide. So middle one that will form a cyclic. So two hydrogen will be replaced with the two nitrogen, and one CH2 group will be added. So mainly 12 addition peak we have observed, and same we have published in the Journal of Pharmaceutical Science. Now, if I discuss about the synthetic peptide 1, so the mass of the synthetic peptide 1 is 750, that is the 5th charge, 4th charge, 3 charge, and 2 charge. All species we have observed by using the orbitrap mass spectrometry, and the theoretical mass is 3748.9465. The ppm error is less than 5 for this molecule. This is the API which we have performed the analysis. So this is a MSMS fragmentation. Now it's quite different for the peptide molecule that we need to select a B or Y ion or A or X ion and B and Y ion, B and Z ion, C and Z ion. So based on that, we can predict the structure of the molecule where MSMS fragmentation happened very easily. So in this molecule, I have highlighted only Y30 and because it's 31 amino acid molecule. So Y30 is 1807 and B1 is 138 around mass unit. Now if I add a formaldehyde and we form the structure of that cyclic 1 carbon addition, that will form a 376012 mass addition. So here also we have observed 543 and 2 charge species that we can deconvolute and form the peptide formaldehyde adduct. And this we have already published. Now, N-terminal histidine MSMS based fragmentation, it tells the B1 ion is 150, that define itself as the addition of 12 mass unit happened at this location only. And Y29, Y30 will remain same. So 1807, that can be clearly seen. So next is we have added a modified NAP test reagent as well as the conformative test for this molecule. And what we have observed the 28.9 mass addition to this formaldehyde adduct, and we have observed the 3789.9366 mass unit difference, and ppm error is less than 5. So it is quite predictable that this secondary amine got nitrosation. It is very quite difficult if I talk about NMR, because NMR require a lot of interpretation while performing a peptide analysis. So what next strategy we designed, that we will take a dipeptide that is the first amino acid and second amino acid. We will perform this structure, and then we finalize the structure based on the NMR for the dipeptide. That can be correlated with this structure. So this is final. This NNO can form in this. So the concern is the formaldehyde, right? So if you control the formaldehyde in your API and in the formulation or any of the excipient, so this impurity cannot be possible. Now we need to control the formaldehyde in our API and the excipient. If you change the source of your API and any of the excipient, then you need to monitor the formaldehyde concentration, because as per my knowledge, ICH M7 says it is around 10 mg per day. Formaldehyde is allowed. Correct me if I am wrong, but it is 10 mg per day is allowable for other than the inhalation project. And in PG, at the BP monograph, I have referred old monograph, but it says that 15 ppm is allowable limit of formaldehyde. So if you can control the formaldehyde with the same limit, then the risk assessment is not required. But if you say that formaldehyde may change, but it is under the limit, that can affect the nitrosamine NDSRI formation. So second molecule is also quite similar with this molecule. I'm not going to present this. This is the GLP-1 analog molecule, so you can refer that slide also. So similar reaction. So this is the HPLC chromatogram for formaldehyde adduct, where 12 mass addition, and this is NDSRI formation. So this is our research group who work for this NDSRI generation. Our department head of the department, Dr. Ravi Picha, associate professor, he contributed a lot in this work. Mine and my research team who is working for this project. Along with this, I have my own research group. So these are the students who are working on the peptide analysis, nitrosamine and extractable leachable studies. Thank you very much. If any question, we will discuss during the panel discussion. Thank you.
Yeah, so, thank you, Dr. Sharma, for the presentation. It's very interesting. So I'd like to invite all the speakers to the panel, and we can do a Q&A. Okay, so I think what we can do is, so I think what we're going to do first is to, how do I say this? To do questions from the audience, and then after some, for virtual questions. So, but if you'd ask the question, please direct it to the panel member, please. That's all I ask.
Yes. Can I start?
Go ahead.
I would have a question for Dr. Sharma.
Can you speak very close to because it's sort of a little bit hard to hear.
Yes, so I have a question for Dr. Sharma. You showed nitrosamine of vancomycin and of a GLP-1 antagonist, right? So vancomycin has a molecular weight of approximately 1,500 and the GLP-1 antagonist even 4,000 or so. Does that let you think about applying molecular weight correction to determine the acceptable intake for such NDSRIs?
Acceptable intake is not as per my purview. I just presented the NDSRI formation, how you can generate the NDSRI to evaluate the risk assessment strategy. So that is a different topic that what we can consider for that. So that is about to discuss. So this is the generation of the NDSRI what I have focused on my.
Thank you.
This is also a question to you, Nitish. Phenomenal work, because obviously these are pain points, dalbavancin, vancomycin, everybody's scratching their head. But what I got very worried about is the last one. These are not nitrosamines, they're nitrosamides, and completely a different class. With the GLP, if you do 2 to the power n, you will have more than 100 nitrosamides there, theoretically. I think we need to draw a line. I'm ardently begging FDA, come out and say that right now we are talking about nitrosamines, show the difference. I had published a paper, but obviously that was, I did talk a lot, that nitrosamides are not nitrosamines. They act differently, and we should not go there. We really should not go there right now. Let us solve the small molecule NDSRIs and then maybe start thinking where we can go with these GLPs. I mean, you have biological molecules with thousands of amide bonds. Do you realize where it will go? So my ardent request is stop, please, stop.
So we respect your opinion. That's your intention. And it's not amide which I have shown. This is the nitrosamide, not amide. Just refer that structure again. I will discuss you in the separate. So the impurity of that peptide, so that can react with. Thank you.
Good morning, everyone. To the presenters, it's an excellent series of presentations, very informative. My question is for Naiffer, and I want to give a little context before I ask the question. It's got to do with the beginning of your presentation about NDSRIs that may not actually be stable. I'll anonymize the NDSRI in the product, but recently in an ANDA application, we received a deficiency from the FDA saying, as applicant, we acknowledge your what we call failure to synthesize justification for a specific NDSRI, therefore not posing a risk in the product. However, we found this reference on the web to a reference standard manufacturer who is offering this nitrosamine. Please acquire the nitrosamine and do the necessary testing. So I summarize, this is the deficiency just recently received. So Naiffer, my proposal is this. The issue is I think all of us may benefit from a third-party arbiter who can be trusted to say, we know this nitrosamine is not stable. You don't have to go, every applicant, you don't have to go chasing after it. Exactly like the ticagrelor case where it's forming a cyclic oxime. It's not actually forming the nitrosamine. We know there are other NDSRIs that theoretically are there but not stable. The calcium channel blockers do not form stable NDSRIs, and there are some other classes. So what do you think about potentially the USP acting as that trusted third-party arbiter to say, we know this is not a stable nitrosamine for the benefit of FDA, other regulatory authorities, and applicants?
Sorry. Thank you for that, Martin. I think that's definitely something that we have been discussing internally. This has also been motivated by that spirit of exchanging information on the nitrosamine exchange. How can the community really facilitate that exchange of that knowledge? I'll be extremely careful using just the list of an impurity in a supplier as the source of that material actually existing. Go deep into really validate if that material actually exists and it's correctly characterized. That's something I will say. Definitely, again, this is something that we cannot do ourselves. We would definitely need help from those folks that have tried and they have failed. I mean, just that information will help us also prioritize some of that work. If manufacturers, if the community is willing to work with us to create that effort and put that information out there, I don't see why the organization will not support that effort. So taking that initiative. Thank you, Marty.
Thanks for that response.
Susan, just because I'm just continuing, is it okay? 1 minute. I totally agree with Martin. I have seen this situation, and I was just telling you the story that it's only after we saw, did our own characterization, we realized. And the internet is flooded with these. And that's also another request to the FDA, not to act like unpaid marketing executive of a small company. We do not know many of these, they are just putting these as theoretical. They don't even know if they can make it. They take a list. So other thing that USP could do is also help characterize, if somebody is having it made somewhere, USP doesn't make it, but if you can use what we were saying, 2D NMR and maybe LCMS, maybe act as a third party, that would be very helpful because half of the time we are having doubts with the standard.
Yeah, thank you for that. Again, I mean, I'll take any ideas, absolutely. That's really where our efforts has been on this space. How can we really help, you navigate all these challenges? The characterization becomes extremely critical when it comes to this substance. So again, be very careful where you're sourcing this material. We have seen labs saying to us, I don't need a standard because I create my own standards. That's fantastic. A lab can actually create their own standards, but they need to know what they're doing, right? How they're characterizing that material. They need to understand the chemistry. It's not about taking the API and putting nitrosating agents on it and pretend that you will get the NDSRI right away with a good purity value. So again, these are things that can be done, absolutely, but again, you need to make sure that all those quality checkpoints are there.
Go ahead, Susan.
Hi, this is Susan Zook from the FDA. I have a question for Dr. Sharma. It was a really interesting presentation about this enhanced NAP test that you're doing. And you talked a lot about molecules that you were able to degrade and form the NDSRI. But I'm curious whether you had tested APIs where you thought that the NDSRI would form and it didn't under these conditions.
So you are talking about that API that difficult to convert in the NDSRI. So yes, but we can say luckily for us and API we have selected till now, we are able to convert in the NDSRI. So my main focus of my research work is in the earlier stage, if the company person know that where my NDSRI peak will get elute in the chromatography. So it is quite easy to finalize their strategy. That's it, what I have presented. So the molecule, if you are facing any challenge to form the NDSRI, you can discuss with me. I can take that API and I will try in my lab to convert that NDSRI. So that what can I do?
Okay.
Thank you.
Thank you.
Yeah, go ahead. And also just want to mention that, in our guidance, it's very clear, if you do the testing, of the, need API under stress conditions, and, you don't form this NDSRI, that could be a basis to de-risk it in the drug product. Because we know that there's some APIs, you won't form the NDSRI. So just to be very clear, when we published the website, we went to the GSRS database, we screened anything that's a secondary amine, excluding, of course, the S9 drugs, and we posted them. But they're all theoretical. It doesn't mean that it's definitely going to form. We know we're starting to get evidence that some that may not form. But and the guidance is very clear about that. You do the stress study, and it doesn't form you. That de-risks it. That's very, very important. That's part of the guidance. So, go ahead.
Yeah, I have a question to probably either of you can answer, Xiang or maybe Zhu. This is about the conversion ratio that you have calculated. So one thing, this is excellent work actually and very helpful. one random calculations, if I was just doing it when the first presentation was going on, like 0.2% if you have an impurity and 0.2% if you consider the max. So that comes out to about 4 ppm. So anything below 4 ppm is definitely, above that is definitely at risk. But the way you presented is really nice. What I'm looking forward is, this is a unique case where you have only the fragment. But you may have cases where you have the API NDSRI as well as the fragment NDSRI. So have you calculated any conversion ratio? Because obviously it's an assumption that the API itself would mostly convert and the fragment may not convert because the amount is less. But have you calculated any fragment ratios in presence of the API, where the API also is a vulnerable amine? Have you any data of that kind?
I think I can take it from my experiment, kind of a view. So we haven't had done any calculation experimentally that you have both API and API fragment. They both, all of them can form NDSRI, and what kind of conversion for API and what kind of conversion for impurity. We haven't done that. So you can look at the data from the API NDSRI. So sometimes you see 0.1 ppm something when you convert. So if you do the kind of a calculation like that a little bit, my overall impression is that from the impurity kind of conversion rate seems much higher than API. Not in the same drug product, but at a different product and impurity and NDSRI conversion rate that we calculate from these two examples.
Okay, thank you.
I had a question regarding the ritonavir example that you had. I thought it was interesting. It's a high dose drug. The CPCA category 1 led to a limit. I think it was 20 part per billion, and your limit of quantitation was above what the AI limit would perform. I guess my question or concern is, in those situations, then how do you actually? I know that you were measuring higher levels, but if your limit of quantitation, you can't detect down to it, how do you then de-risk those NDSRI related impurities as well?
Yeah, thank you for the question. We actually had a little bit of discussion with some of the attendees during the break. So the reason that we have this relatively high limit of quantitation is because the sample we used for validation, for the accuracy, we do the spike recovery. And then the samples we use as a placebo, kind of do this spike recovery. There's already high level of the fragment NDSRI in it. So really we cannot spike in very low level to demonstrate that we can reach the LOQ kind of to the limit, because the variation there will not allow us to do that. So in order to show that our method really can reach that low kind of limit, we needed to have a placebo that really, doesn't have much of the NDSRI present in order to do the experiment. It's just due to the lack of such a placebo in our method development, we could not show that we can get to that LOQ. That makes sense?
So in essence, if you did alternatives, you think you would be able to get down to 20 part per billion, or?
Yeah, I think based on our experience analyzing different NDSRIs, I think the sensitivity usually is not quite an issue here. Yeah, thank you.
So I'm not sure if you answered this question yet. So the examples you gave spoke to specified, or at least secondary amine impurities and tertiary amine drugs that are known. So there are countless tertiary amine drugs that do not have any secondary amine impurities. How do you perceive the risk associated with tertiary amine drugs in a solid oral dosing situation which do not have any secondary amine specified or unspecified impurities that just aren't known?
Yeah, that's an interesting question. I guess that we haven't really looked into an example like that. And we are like, welcome any thoughts that any drug that that a tertiary amine, but not really have like a secondary amine impurity present.
Yeah.
Yeah, just like here's assumption is that secondary amine is more likely to have more nitrosation than the tertiary amine.
Yeah, I just want to say, just because it's unspecified doesn't absolve you of that responsibility. So I want to be very clear. So I think the point of that talk was, there are a lot of drugs that could have secondary amine impurities, and people sort of said, that the risk would be very low for NDSRs. We just did the simulation, sort of saying that under certain circumstances, especially high MDD, and the secondary amine is the CPCA1. Those are sort of high-risk case scenarios. You should consider that you should consider that during your development. You shouldn't sort of say it is a negligible risk. It's probably not. It's something just to consider. But then conversely, if it's a very low MDD drug, CPCA is 4 or 5, probably the risk is lower. It's just something just to bring to the attention. So we shouldn't. Secondary amines can form. And we have those examples. The ritonavir example, the triplet. It's indicate those can form. So.
Actually, almost as a continuation of what you were saying, Andre, I was just going to say that tertiary amines do nitrosate. I mean, that's like my PhD advisor, Dr. Lepke, did his PhD on that. Some of them nitrosate quite fast, but some of them do it slowly. Let us remember that a drug product is a reaction vessel where the reaction runs for 2 years. What is a reaction that you let it go for 2 years and you'll not see a few ppm or ppb? So even if you don't see from a tertiary amine doesn't degrade to a secondary amine unless it's in the liver and it gets oxidized. And in many cases, you don't use a secondary amine maybe in the synthetic process. So it may not be an impurity, and yet you will see. I mean, in my 12 years of PhD in NCI, I have never seen a secondary amine that doesn't have a nitrosamine. The biggest evidence is triethanol nitrosamine, which triethanol amine, which is very rampant in topical dosage forms, always has NDELA, and that's in cosmetics. And actually, triethanol amine, if you see CFAs, has a control of Nadella. So I just wanted to say that, it's a very slow reaction, but it happens, especially when you leave it for 2 years. So whether there is a secondary amine or not, it should be looked at.
So just wondering, Dan, is anyone from the virtual any questions you have?
Hi, so we have a couple of questions from the virtual audience. So starting off with taking into account the difficulty of getting NDSRI reference standards, especially for new chemical entities, if analyzing a drug product to support a risk assessment, is it possible to quantify the nitrosamine using the response factor in electrospray of the API as a surrogate for the NDSRI?
Oh, go ahead. Okay. So the question is that whether can use a surrogate to do the quantitation,
right?
The response factor. But then how you calculate the response factor, you still need a reference standard to calculate this response factor. So how do you know the response factor?
Well, you don't. You're just assuming it's the same as the...
Oh, yeah. So then...
Is that a reasonable assumption, I think?
Right. I think if you wanted to have an estimation ballpark estimation, that's probably okay. But if you wanted to have a reliable, accurate quantitation, that may not be acceptable.
Okay, thank you.
This is the question regarding the work by Dr. Sharma, but not really directed to him, but to the probably FDA. This is, as per his data also, we see that in terms of the original NAP test versus the modified NAP test, you find that there is more amount of nitrosamine which is being formed. But we have to also consider that the original NAP test is more realistic in terms of the actual solid state reactions that may actually happen in a product. So not only the modified NAP test, in these days we see a lot of fancy mechanisms or fancy chemicals being used to produce a nitrosamine and NDSRI, but are there also indicative that this is not realistic, which may actually form in the product? So do we should consider that part? It's not really a question, but a thought.
So I would like to answer this question. So it's not that compulsory that you need to perform the modified NAP test. Yeah, you what is your requirement that you first find out that your product is forming a nitrosamine or not in the real product scenario, right? So what you can do, you can perform all three tests simultaneously. You can check the characterization, and first you can evaluate that which strategies suit you, where you have the minimum amount of nitrosamine that you can finalize. So my strategy is quite for applicable for the initial stage of the product development, when you are more focusing about the product strategy, product development stage or product finalization. Right? Once you finalize your strategy, excipient, process, everything, it's quite difficult for the company to change their strategy. So because at the initial stage, you are not able to you are not able to separate the nitrosamine, right? So it's very initial. So what you do, you perform the force degradation study, like to prove the synthetic and the degradation nature of your API of your impurities. So similarly, you can adopt this strategy, and you can have a method at very initial stage. Later, yes, you require a reference standard, working standard, which is fully characterized. So by using this method, wherever you find the maximum amount of NDSRI generated, so you can isolate that and you can characterize that. But yes, the orthogonal approach should be applicable while you are characterizing the nitrosamine impurity standard.
Yeah, Miss, I fully agree the analytical part of it, the quantification part of it, but I'm talking of the actual formation possibilities. Means I have seen mechanisms where you are blocking some of the groups, nitrosating it, and then unblocking it, which is not really going to happen in real life situations. So that was the thought basically. Thank you.
So there are a few questions about potential instability of nitrosamines, including in HPLC diluent or under the HPLC conditions. Is this something of concern? Is this a common problem for nitrosamines?
I mean, I would say, as any analytical method, these are variables that we need to assess. The stability of the material, the stability of the solutions, how they're going to behave in the instrument as it undergo transformation during the analysis. Again, I pointed out the example of the ranitidine. Very early, we start seeing formation of that nitrosamine because the temperatures on the injection port. So again, these are all parameters that we need to consider as part of that analytical development strategy.
Thank you. And kind of a related question, there's also a question about what degree of characterization data is needed to authenticate an NDSRI. So would mass spec and proton and carbon-13 NMR be sufficient?
Whatever is needed to make sure that what you get on that vial, it is what it is.
Okay.
Again, I have a question now sort of combined for Jan and Shane's presentations on both the experimental evaluation of conversion factors and then the modeling based on that. Shane, you said something towards the end of the presentation that showing that the modeling showed can be a significant percentage of products that due to fragment NDSRIs could be on your high risk category as defined. And you did indicate that maybe that may require the precursor to be controlled at levels lower than Q3A. Is there a consideration within the FDA on further conversion studies to establish, let's say, I mean, I realize there can always be exceptions, but in general, that conversion in actual drug products doesn't exceed a certain factor, like never see an example higher than 2% or something like that. And the reason I say that is that if it was really important to do this work and to try to get a handle on it, I think that was a great idea to pursue. But if we just have at this point a very limited amount of information on the extent of conversion for fragment NDSRIs, that may cause a consideration to push a lot of what have been historically been Q3A controlled impurities to much lower levels. And the impact is potentially, I think, on industry. We're already tying up all our LCMSs and buying more to test the NDSRIs themselves. We would now be potentially moving into the space where we're using LCMSs to test precursor non-nitrosamines. So I'd appreciate your thoughts on that.
I just from my point of like experiment or like that, nothing about like that side. So I agree that the data we tested still kind of like a limit. So yeah, it may be that we need to go out to get more different scenario and to get more understanding of the extension rate, that conversion rates that could up to what and like that. So that's kind of like a comment from my side.
Thank you.
I can add a comment to this. So so I just want to be very clear. We're not trying to tighten ICH QT thresholds. Okay, to be very clear, all we're just saying is that and there was one slide that he did a simulation for all the drug products in FDA. We did at 1,200 impurities. It's just representative. But he also in part of that he did a slide for just one impurity. Like just one. And that's what I think what we want people to think about is that you should consider this. And there's sort of two ways to resolve this problem. Okay. One, if it's a high risk scenario, high MDD, low CPC, which there's sort of two ways. One is to limit the extent of conversion. There are tools to do that, okay? We know that the antioxidants or the pH modifiers, that's one tool. We're not saying that you need to, limit, the nitrosamine to extremely low, not the nitrosamine, the precursor to extremely low levels. If you do that, you may not need to do that if you use the conversion approach. Or the alternative, if you decide not to reformulate, the alternative strategy is to work with your API supplier to have a very stringent limit on the precursor. I mean, there's sort of two ways to resolve this problem. So we're not saying so I want to be very clear. We're not saying universally we want everyone to have very, very low levels of secondary, impurities. I think those would be very case by case, basis. And maybe in some cases it may be needed for your specific product, but not universally. I don't know if that makes sense.
Yeah. Thanks, Andre.
I just wanted to ask another question. I saw a lot of information. The focus is a lot on the solid oral dosage form. Did you try to look into other dosage forms? Because they exist as much as anything else. And, it would be interesting to also know that if there is a transdermal or an inhalation or, products like that, how this changes, especially the impurity. I'm like extremely curious that how it would act in a different kind of a dosage form. So just curious.
Yeah, that's a very good question or good, interesting comments. That different dosage forms, they may have the different rate or whatever we call it. So, and a little bit kind of like assumption here is that we consider like a solid dosage form may have the worst case of a scenario. And then the oral solution, usually their pH is kind of a bit controlled, and the number of the excipient in it may not be that much. So relatively, it may not generate high NDSRI like that compared to the solid dosage form. And we do have a little bit of limited data to show that like a comparison of the dosage form, like tablets to oral solution like that. And then oral solution did have some like a lower kind of like NDSRI compared to tablet form. Okay.
And I just want to add one point. You may already have a lot of this data from the API. I mean, remember, in pure, the API is not, so remember that, so think about, we're seeing a lot of these NDSRI in solid, obviously high propensity, but then, look at your analysis of, liquids and see what you did. I don't know if, probably the answer is not going to be significantly different for impurity. Impurity, secondary mean is secondary mean. They don't care whether it's the API. Yeah. 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 Yeah.
We are looking at a cytochrome P450 mediated immunogenicity and carcinogenicity, and we have a significantly lower absorptive capacity. Is there any rationale for assuming that we should use 100% absorption for, say, a topical NDSRI?
Yeah, I just wanted to do, that's a farm talks question, which I can't answer, I'm sorry. So I can't answer that question.
So I have a question for Jen. I can't stop thinking about the manufacturer C for your amitriptyline product, because apparently your data show that their level of NDSRI is much lower than the other manufacturers. So I was wondering, as risk mitigation, right, would take if you change a wet granulation process to a dry granulation process, would that help mitigate the risk of NDSRI formation or growth during drug product manufacturing, especially after hearing one of the talks from yesterday, right? The water can facilitate the NDSRI formation during drug product. So I'm just curious about your thoughts on, whether taking water out of the drug product process would help.
I think I may not be in a position to talk about that, what kind of like manufacturing process maybe help with the NDSRI formation, because that's not really, I'm not really dig into that, and I cannot disclose like a manufacturing C for you to look at into the manufacturing process to see why they have like a lower kind of like percentage of the NDSRI in the drug product. So maybe someone here can take that question about the kind of like what kind of like a manufacturing process have higher risk or low risk of like could affect the NDSRI formation. And
I'm really asking to start a conversation. So anybody, if anybody has any thoughts or input to share, right, I'd appreciate it.
No, I was, Ian was here. I don't know if he's not calling in, but he wrote he gave the presentation, based on purely theoretical grounds. It could help, but again, I think you should confirm it with experimentation, but from a purely theoretical standpoint, based upon what you presented, yes, probably. But again, you need to confirm it with your data.
Okay, thank you.
So it's What time is it? It's 11:20, so, I guess, we're done with the questions. I want to, thank everybody. And, thank you all for this session. So, and what am I, so I guess, I'm supposed to give some concluding remarks? Okay, I'm going to try, I wrote something very brief to give some concluding remarks, about this presentation, so I'll just try to summarize, something very brief here, and then folks can go to lunch. So, let me try to write a summary. So remember, what was the title of this session, for the 2 days? I think, and we accomplished our goals, I think, was talking about the risk factors of formation, there was a lot of talks on that. There was the talk from, we always think about, nitrites in formulations forming NDSRIs, and that's probably the most prevalent. But, there was a very interesting talk from Dr. Joe Seligman, about the enoxamer. So it adds sort of this additional dimension of a failure mode that should be considered during your risk assessment. That's another obvious failure mode that can result in the formation of the NDSRI. And then also we heard talks from Martha Essendahl discussing the risk factors for certain excipients, particularly the HPMC, hypromellose on the formation of NDMA in extended release products. We also had a talk from, Dr. Nitish Sharma. He talked about the modified, NAP test to discern the secondary amine drugs. And I think that's a very important test to do these sort of type, very stressed, modified, tests, because if the test indicates, that you're forming the NDSRI, you probably need to consider that in your risk. You can't completely de-risk it. But if you do all these strenuous conditions and you're not forming an NDSRI at all, and we have seen those cases. I mean, I've seen cases which you have secondary amines. No matter what you do, you will never form that NDSRI. And that is one pathway, and we discussed that in our guidance for de-risking. So that's another something to think about. And then we heard from, Dr. Ian Ashworth on, he developed a very nice mechanistic model, kinetic model based on first principles, very elegant. I admit it sort of overestimates the risk, but it could be used as a tool, like you said, like the liquid, and to ascertain the risk of nitrosation of drugs in formulations during the course of your development strategy. And, we had two very interesting talks about, API fragments of NDSRIs. Like I said, our focus initially was on the API, and rightly so, because the API is most of the product. The API is much less. But, I think Dr. Jingyue, presented some very nice studies showing that for certain products, these NDSRIs from impurities do form, and they can form in appreciable levels, and we shouldn't neglect them. And then Xiang, he did a very wonderful modeling study using a repertoire of impurities we have in our GSRS database, showing that we definitely cannot ignore these things, that especially those sort of high-risk scenarios, high MDD, low CPCA, this is something that you can't just ignore and just say there's no risk. It should be evaluated. That was what was presented, and probably there'll be more discussions on this. And finally, in terms of risk, we heard talk from Dr. Daniel Berger on multiple nitrosamines. He elegantly showed the various case scenarios in which you can form it. It could be an API, it could be purity, it could be fixed dose combination. He showed those scenarios, and they're going to happen. So that was a nice thing you need to think about it. But also he also talked about our evolving regulatory paradigm of how we're going to control these nitrosamines. I remember when we did this before, we would say the most stringent nitrosamine, but we realized when you have nitrosamines of different orders of magnitude, it may not be feasible. So we evolved our paradigm, using the flexible approach to ensure that it's no excess of 100,000 cancer risk. And then, of course, when you do the risk assessment, if there's a risk, you need to do confirmatory testing. And we had some very nice analytical, presentations. We heard some very nice talks from Naiffer Romero from USP, talking about, And I think you pointed out some very important points. Of course, the importance of a reference standard. I think it's very important to have a valid reference standard, and it's true, there could be some reference standards that are not what they purport to be. So you have to be very, very cautious about that. And of course, in developing your analytical method, the specificity matrix effects and other artifacts that could either overestimate or underestimate impurities testing results. And we had the talk from Dr. Guozhang Zhao, on some of the challenges of quantitating some of these nitrosamines in the model, the nitrosoketamine. I didn't realize it was very, very difficult, but I realized, and now I understand some of the challenges, especially in that case scenario, and how was analytically resolved with the solid state extraction. And then, finally, we had the talk from Dr. David Curie. It was very, it's talking about, when these NDSRI forms, we have strategies to resolve them, and he provided the talk for the metformin, in which used antioxidants. And I thought that talk was very, very interesting because, we compared it with bumetanide. We had 3 antioxidants we used. It was ascorbic acid, caffeic, and ferulic. There were 3 at once. But in case of metformin, in the case of bumetanide, ascorbic acid was the most superior by far, no questions. But when we did it with metformin, the other drug, it was actually the worst. So it really tells you that as you're mitigating, it's not like the one magic, antioxidant that you can use. You have to, it sort of depends on your particular drug and your particular formulation. And also those studies also did reinforce the important point that pH modifiers really, suppress dramatically, probably even better than antioxidants, the formation of NDSRI. So there are definitely pathways to resolve these issues. And the last thing I really wanted to really mention is that I was in this conference 1.5 years ago, and I think we made a tremendous amount of progress. I mean, when we presented in 1.5 years ago, all the nitrosamines were 26.5. I remember that. But now we have the CPCA. So, they're not all 26.5. Now we have, levels that can range to up to 1,500. That was articulated in the rail guidance. We actually posted, there was some, I think about the NDSRI, there was some confusion about which APIs people should pursue or not pursue. So what we did was we posted all those APIs, that people should consider as a possible risk. So that was also a big development. We updated our quality guidance, to better reflect when the original one was written, was mainly focused on small molecule nitrosamines. We didn't really think much about NDSRI, so we had to update it to that time to reflect that. And we also involved our paradigm for total nitrosamine control. And, and there's going to be a talk in the afternoon about, we know that antioxidants can work, and we wanted to make it easier for sponsors to incorporate the strategy. So there's going to be discussions on this biowaiver option to mitigate, so you don't have to avert these, costly in vivo, PK studies. So all I can say to you is that I think a lot has been accomplished in the last 1.5 years. And I think these very fast, if you think about it. And we still have certainly more to do. I acknowledge that. There's a lot more needs to be done, but it really shows that from the regulators, industry are really working, together very, very, to overcome this very, very complex problem. And hopefully when we have the next CRDC, we'll have, more solutions. So thank you all for your attention.